好，咁呢個 video 咧就係想用嚟講解翻整個 genetic engineering 嘅過程啦。咁相信喺上堂裡面咧，大家對嗰個整個 overview 都有個唔錯嘅理解㗎啦。So 7 key steps are involved in the process。咁我特別想介紹翻幾個詞語，等陣喺討論裡面你會容易啲理解先。咁當然 start with 一個 gene of interest 啦，係咪？咁我哋講過啦，佢需要有一個 join into a plasmid to form a recombinant plasmid。咁但係你要諗一樣嘢，就係點解需要個 plasmid 咧？因為我哋講過，如果佢呢個 short 嘅 gene of interest 就咁走入去個 bacteria cell 裡面的話咧，it will be broken down by the bacteria because it is foreign。所以咧，我哋就需要將呢個 gene of interest 連結落去嗰個 plasmid 裡面，成為一個 recombinant plasmid。好啦，而家所以有兩個新嘅詞語要介紹啦。咁我哋想講翻呢兩個 DNA 嘅角色啊，所以一個 plasmid 咧 ，it is acting as a vector， 一個載具、運載嘅工具咁解。while the gene of interest is acting as an insert， 好似一個插入去個 plasmid 嘅，即係個 vector 嘅一個短片咁樣。咁所以有時我哋會交替用呢啲括弧嘅詞語嘅。咁個括弧嘅詞語就係代表緊佢哋嘅角色啦嚇。OK， 咁我哋喺討論嘅過程裡面，我會不自覺可能呢啲詞語互換，咁所以你要留心啦。呢段片最重要嘅目的咧，就係想你理解點解我哋需要一個 step 4。step 4係代表咩咧？即係話，當你已經將個 recombinant plasmid transform 咗 into the bacteria，then right the cell number of cells is very low. You want to grow and reproduce these cells into a great population so that you want to get huge amount of proteins, right? But there are some inherent problems, intrinsic problems in step 3 that makes the whole process not so ideal. And if you don't solve that problem, the eventual amount of protein that you can get will be extremely low. So let us dig into step 3, see what the problem is, and think of a way to solve this problem, and that solution would be step 4. Okay, so this is a very, very important video, very, very important learning point, always asked in DSE. Okay, so please take out a blank paper or a single line paper to draw together in order to understand the problem during the transformation and the solution. So I'm going to draw pictures to let you understand. So I will pause my video when I won't pause during the process. So when you want to follow the progress, so you pause the video at any time to complete your drawing before you go to the next step. Otherwise, if you keep drawing and you can't listen, you won't understand. Okay? Everything that I draw will be in this whole screen there, so you can 你自己預位啦，你自己因位咯。So please pause the video and draw these things first. So now I continue. Once a recombinant plasmid is made, you want to transform this plasmid into the bacteria. All right, let me change the color a little bit. But have you ever think about this is too ideal? right? The bacteria, there are two possible outcomes after this transformation. Now I tell you that the transformation process is not perfect. The bacteria may successfully absorb the recombinant plasmid, or they do not. So can you now draw two types of bacteria to represent these two possible outcomes? Pause the video and complete your drawing before you play the next part. Alright, I'm going to continue now. If the transformation is successful, the bacteria would have the one or multiple copies of the recombinant plasmid present in the bacteria. While if the process is unsuccessful, then there will be no recombinant plasmid in the bacteria. So let's give them a name. This is bacteria with recombinant plasmid. In other words, they are the transformed bacteria, while this is the bacteria without the recombinant plasmid. And that is a non-transformed bacteria. Now in the exam, you know, in the video now, I'm going to use the term transformed bacteria and non-transformed bacteria. But in the exam, it's better to write the full description above. Okay, bacteria with or without that plasmid. Obviously, transformed bacteria is going to express the gene of interest and produce the protein that you want, while these bacteria, non-transformed bacteria, will not do the surface. So the first thing that you realize here is that what we are talking about is the transformation efficiency. And this transformation efficiency is not perfect. It's not 100%. So what can you imagine? What's the problem if we let it go on for step 5 and step 6? So pause and think a little bit, and how it was going to affect the yield. So let me give you a scenario. All right. So let me say the transformation efficiency is 70%, which is obviously already very high, higher than practical. But would you imagine how is the impact on the amount of protein that you can get? So pause and think. All right. So you may guess that, all right, so I think obviously everyone will understand that you will get less protein because these unsuccessful bacteria, non-transformed bacteria growing inside this flask, this culture medium, they are not going to produce protein. So when these two types of bacteria, transformed and non-transformed ones, when they grow together, it obviously is going to reduce the protein amount that you can get. But maybe you are thinking, all right, so a 70% transformation efficiency would cost you a 30% loss in the protein amount. But actually, the impact would be far greater. And I would say, if this problem is not solved, even a 70% transformation efficiency would result in nearly very, very little protein produced, nearly none. Can you imagine why? So let me give you a hint. is related to the idea of natural selection. Obviously, this is not, well, yeah, it's related to natural selection. So can you pause and think about it? Think about what happens here. Can you imagine what happens so that the protein production would be so heavily impacted? So I'm going to review the answer. I hope you have paused and think. It's very important. Okay, so let's go back to the drawing here. So now let's say the next step is to put these two types of bacteria together. Remember, you don't know, right? In the bacteria, you can't see which one is transformed, which one is non-transformed. So when they grow together in the culture medium, so what's the problem? So the problem would be that now maybe you think, all right, so there's just a little bit of non-transformed bacteria in the flask. So what? Most of the bacteria at the start is the transformed ones. But think about the transformed bacteria is producing hemoglobin. Also remember that when you have done the homework, you should know that there are numerous copies of these plasmids inside the bacteria, so they are making numerous hemoglobin inside one cell. Think about the workload. So they need to sacrifice a lot of energy and a lot of materials, say amino acids, building a protein that is useless for them, for their survival. While the non-transformed bacteria, they can reserve all the amino acids, all the glucose to make their own proteins, release energy for their own cellular activities. So which type of bacteria would grow better? Now obviously you would now realize that the non-transformed bacteria would grow better. So now also remember that the bacteria is reproducing quickly. They are reproducing every single minute, let's say. And this culture medium, we are going to grow it for at least 1 or 2 days. Imagine what will happen according to natural selection. So when they grow together, at the start, most of them are transformed bacteria, alright? But since they are wasting so many energy and raw materials for making hemoglobin, they can't survive well. On the other hand, the non-transformed bacteria will be surviving much better. So, they can reproduce much faster, and the offsprings that they produce obviously would not contain the recombinant plasmids as well. So the proportion of this non-transformed bacteria would be increasing and increasing and increasing. And imagine repeating this for 1 or 2 days. That covers already millions of generations of bacteria. Each generation, the proportion increase. So eventually, even though you start with a 70% transformation efficiency, but after 1 or 2 days, eventually this flask will be mostly dominated by the non-transformed bacteria. And what's the problem of this? then we say the protein yield would be all greatly decreased, and it will be heavily impacted due to natural selection in the culture medium. So how can we solve this problem? Now, the first solution that you think of, maybe, all right. So let's find a way to improve the transformation efficiency here. Let's all right. 70% is too low. So let's find a way to make it 80, make it 90, make it 100. Oh no! You can't make it 100. The 99, let's say. Is it a viable solution? Is it a workable solution? So think about the natural selection process. Even you raise the transformation efficiency to a very high level, as long as it's not 100%, these non-transformed bacteria will eventually dominate. So it can't work. And well, not to mention that this is a very random process. We can try to make the transformation more efficient, but it's quite difficult. So how to solve this? The key to solve this problem is find a way to distinguish these two types of bacteria, the transformed one and the non-transformed one, and find a way to eliminate the non-transformed bacteria before. Now remember, you have to do this before you let it grow into a large population. Alright? So how can we solve this problem? Let me give you a hint. The hint is you can refer to a plasmid in which they usually contain an antibiotic resistant gene in the plasmid. So if you want, you can pause the video and think about the details how to solve the problem before you listen to my solution. Alright, we're going to continue now. So now it means in these transformed bacteria, they also contain this antibiotic resistant gene. So what you can do is when you start transferring these bacteria into the culture medium, you add antibiotics into the culture medium. When you add the antibiotics to the culture medium, what will happen? You can imagine the non-transformed bacteria without the recombinant plasmid are killed. While the bacteria with recombinant plasmids are not killed because they have the antibiotic resistant gene. In the plasmid. So therefore, no matter, so you can now imagine, no matter how low the transformation efficiency is, now in reverse, is even though you only get 10% transformation efficiency, even though you start with a very few amount of transformed bacteria, that's not a problem anymore. Because once you add them into the culture medium, these non-transformed bacteria would be killed. So all the bacteria that you have after 1 or 2 nights of growing would be all transformed bacteria. Amazing, isn't it? We call this concept selection. So by adding antibiotics, the transformed bacteria will be selected. So what's the advantage? It can greatly improve the yield of protein. So step 4 is to select. Okay? So once the recombinant plasmid is transformed into bacterial cells, the bacteria with recombinant plasmids are selected by antibiotics. So when you grow and reproduce, most of it, all the bacteria that you can get would be the transformed ones. Got it? So I hope by this video you would understand the following ideas. Understand that transformation is not 100% efficient, and that would result in that some bacteria would be non-transformed. These non-transformed bacteria would heavily reduce the yield of protein. Second idea, to solve this problem, a selection marker like antibiotic resistant gene is utilized. These selection markers are present in the plasmids so that we can add antibiotics into the culture medium to kill the non-transformed bacteria so that only the transformed bacteria can grow and reproduce. Third idea, what's the ultimate goal of this? The ultimate goal is to improve the yield of protein. So please, use what you learned to fill in this question. You can leave this blank. We will talk about this in the class. Okay? So now sorry, to, one more thing. I'm sorry that my answer forget to answer this question. A factor is needed so that otherwise, so you can say it's needed the gene of interest into the whole cell. But also remember that the gene of interest alone would be broken down by the whole cell, in the whole cell. So I'm sorry that I forget to write this answer, and I hope you have a good understanding for this part. Now, one more, the last reminder before I end this video. This is just an example. I mean, this solution is just an example, but it is the most commonly used way to select transformed bacteria, even in the lab that we work in, the research lab or the science laboratory. So this is, but there are other ways to solve this problem. So these other solutions may be present in the test or exams. So it's very important for you to understand the rationale behind. The rationale is that we need to find a way to tell the difference between these two. Okay? In our story, in this story, not only that we tell the difference, but we kill them. Alright? But there are other ways. Say, for example, if I can make this to have color, let's say, let me go crazy. Okay? And this is not. So maybe I can visually take this bacteria out and put it in so that there is no problem. So there are so in this case, there need to be another selection marker, not antibiotic resistant gene, so maybe another gene that can make the bacteria blue, let's say. So there are other ways of solutions to solve this problem. So it's important for you to understand the rationale behind that. We are trying to tell the difference between these two. Find ways to tell the difference. Yeah. So that's all.